Hi, I'm Allison Campbell, and I am from the Pacific Northwest National Laboratory, and I represent the Earth and biological research that we do here. I want to talk today about scientific exploration and what it means as scientists to probe new frontiers, new scientific environments, and new discoveries. And what I want to focus on today is plants and plant sciences. Think about your own scientific exploration and how you push the boundaries of discovery.
So let's talk about plants. You guys are involved with a really interesting project where you're studying plants in a very unique environment. So tell me about the project and what you're doing and why you're doing it.
So the project's a collaboration with Norman Lewis at WSU, where we're taking Arabidopsis plants and wild type and different mutants that have a diminished capacity for producing lignin, which is the polymer in plants that make them stand up straight. And we're growing them on the ground, but we're also growing them on the International Space Station. NASA produced what's known as the Advanced Plant Habitat, and so it's a growth chamber that controls every aspect of plant growth: humidity, temperature, light, water, and monitors it from the ground, both on the space station and the ground. So every aspect of their growth is identical except for gravity. And so we're actually able to compare the plants in a normal habitat and in microgravity.
Lignin is the
structure?
biopolymer in plants that give it its structural integrity so that it can grow up against gravity and transport over long distances nutrients and water.
Okay. And any results so far?
So
what are we learning about all of this?
So what we've seen is that there is a phenotype difference on Earth and in the space station, whereas on Earth the plants seem to just grow straight up and at a random pattern, and on the space station we thought they'd go straight up because there's no gravity, so they would just kind of float up. They don't. They grow to the side and they come out. All the stems are coming out in a counterclockwise pattern, and so there's a pattern. There's something going on that's making this pattern in the plants on the space station.
Plants naturally, both on Earth and in space, they oscillate naturally, and that's called circumnutation. And it was a term actually originally coined by Charles Darwin in 1880. There's evidence that this circumnutation is also coordinated somehow with gravitropic sensing of the plant, as well as circadian rhythms. And so it'll be really interesting in this experiment to tease out what's going on. And we hope to do that by analyzing the molecular level data.
Oh, you took the plants out of their native environment. Did this project take you guys out of your native environment in any different ways?
It did for me, in the standpoint that when you normally do an experiment, there's a certain amount of care and a certain amount of planning that goes into the experiment. I've never experienced the amount of care and planning that went into the space station experiments. It was 3 years in the making to build the advanced plant habitat, to test it, to do the ground control. And I appreciate it because obviously sending things to the space station is expensive. But the sheer amount of planning that went into this is just astounding. And most people don't see it when they see the final results.
So what's next for you? Now you've gone to outer space. What's the next new environment that you're going to explore? Any ideas?
Wow. I mean, building upon the NASA, I think, we need to go to Mars. We need to make potatoes on Mars like they did in The Martian.
That's right.
But that was actually one of the greatest things for me, for working with the NASA folks, is it just really expands your mind.
I mean, we make fun of The Martian, but the reality is as explorers, we need to think about going out and about. So I think that this is a nice step in that process.
But bringing it back down to Earth, understanding the deposition of lignin into plants is also very important, because lignin is the biopolymer that's recalcitrant for biofuels. And so this is going to give us a lot of information on how we can better process plants on Earth, how we might be able to better engineer plants on Earth so that they can be used as better biofuels to take care of the planet we're already on.
I mean, when you started 20 years ago, did you think you'd have an experiment on the International Space Station?
No.
Never. So there you go.
So exciting.
All right. Well done. Thanks.
So what did you think? What did you learn? What I learned is there were two scientists who really stretched outside their boundaries to explore how plants adapt and grow and function in a new environment, such as microgravity. What I learned too is that they open their mind to new possibilities of doing things, new collaborations, and new ways of conducting and planning science. I'd like to hear from you. Please use this YouTube channel to add your comments and thoughts about the things that you're doing in your own laboratory and in your own professional lives. What are you exploring that's new? How has it helped you grow? What new discoveries have you found? Let's have a dialogue. Let's talk about it, and let's have fun.